And so now our work is to really do this next step, how we get from images of the brain over time into a measurement of neuronal activity that is typically presented as part of a neuroimaging study. And then the talk after that, just again to preview, is that final bit of inference. OK, so here's where we're going to start. So I think a good place to begin in thinking about the properties of brain imaging data, right, and the ways that they're analyzed and the sorts of statements that can be made about brain imaging data is through the lens of what we're going to call a two systems model. This is something that we'll touch upon now and in the next lecture. So what's the two systems model? The idea is that in neuroscience experiments, right, neuroscience neuroimaging experiments, one is often interested in looking at the relationship between some aspect of behavior or cognition or consciousness and relating that to neural activity, right? What I'm referring to in this slide as system 1. This is really not going to be any help at all. So system 1 is on this side, and it's basically the system, the thing that relates input and stimuli and instructions to the subjects and their hopes and dreams and emotional states, and how those things are related to neuronal activity, okay? So I refer to this as a system. What's a system? Well, this is a term borrowed from engineering, and a system is anything that you wish to study that takes input and provides output, okay? So lots of things are systems under this kind of construction, like a car is a system, and you can think of approaching a complex system like a car from different input-output states. Like you could imagine the input might be the pressure on the gas pedal, and the output is the speed of rotation of the tires, right? But you could imagine that there are other systems and ways of measuring that. All sorts of things are systems. So in the context of neuroscience and psychology applications of brain imaging, the system that is usually under study is the cognition consciousness system. Input are instructions and stimuli, right? And constructs about the emotional mental states of the subject, and the output of the system that you wish to measure is neural activity. Now, of course, we can't just directly measure, don't worry, I haven't gotten very far. The output, we can't just directly measure neuronal activity in human subjects. Of course, that's why we have neuroimaging. So that neural activity has to be viewed through the lens of a neuroimaging method. And that's the second system, right? The second system, which is what we spent our first lecture talking about, is the system that takes neuronal activity and turns it into a brain image, okay? Where the input is neuronal activity and the output is a brain image. And it's worth pointing out that these two systems have very different properties, right? So in a few minutes I'm going to make the claim that this second system, the thing that takes neuronal activity and outputs a brain image and a brain imaging signal, that system is pretty close to a linear system, right? And what a linear system means is that if you double the amount of neuronal activity that comes in, you pretty much double the brain imaging signal that comes out, right? And if you shift the neural activity in time so it's a little bit later, you shift the bold imaging signal, the measurement you make, in time by the same amount, right? It is a time-invariant linear system. Engineers love time-invariant linear systems. They're very lawful things that are easy to study. And that's to a first approximation, that's the property of the second system. That's great. These are not things that are at all true about the cognition consciousness system, right? Okay, so if you double the number of things that somebody needs to remember, that does not mean it's going to double the amount of neuronal activity in some area. If you delay the time that they need to remember something, it doesn't mean that you're going to get the same neural activity 20 minutes later when they've had a cup of coffee, right? So properties of this first system are very different from the second system. In fact, it is the lawful properties of system number 2 that allow us to study system number 1, right? Because we measure these brain images and we work our way back to statements about neuronal activity, right? So because this second system is rather lawful, because it has these properties of being a time-invariant linear system, which I will spell out in just a moment, from a given brain picture, you can make some pretty good statements about what the neuronal activity was that gave rise to that state, right? You can link those two things pretty closely and work your way back to neuronal activity. And then you're ready to engage in the neuroscientific enterprise of trying to relate a measure of neuronal activity to this cognition and consciousness and stimuli and all the rest, right? Which is the much harder job, okay? So two systems. So we're going to say just a few more things about this system number 2 and what its properties are in time and space. So let me give you a sense about how these sorts of measurements are made and how we sort of actually conduct a bold functional MRI experiment. By the way, bold, I haven't defined that term yet. blood oxygen level dependent. Right? That's the acronym, the acronym. And so that name comes from the main source of the imaging signal that we measure, which is this variation of oxygen in hemoglobin in the blood. Right? So blood oxygen level dependent functional magnetic resonance imaging, bold fMRI. Okay, so this might be a typical kind of bold fMRI image. This is one axial slice through the brain, and actually that image, of course, is made up of lots of little points, right? So just as a TV screen, if you get in really close, you can see the tiny pixels that make up a picture. In a bold functional MRI image, it's also made up of tiny little points. In this case, they are called not pixels, but voxels. And the reason they're called voxels is because they have a volume element to them, right? Because each point of measurement is a cube of tissue. And in typical application, that cube of tissue is about 3 by 3 by 3 millimeters. Though actually in modern imaging techniques, we're starting to move a little smaller, more like 2 by 2 by 2 millimeters, right? It turns out that you can do the calculation of, you know, how many neurons are in the brain, and then, you know, how big a cube is and how big the brain is. You can do the math. Turns out that each cube, if you have about a 3 by 3 by 3 millimeter voxel, contains about 630,000 neurons. In fact, if you make your cubes half that size, right, so if you go 1.5 by 1.5 by 1.5, that's, by the way, not a halving of the size, but a 1/8 of the size, because we're dealing in volume. So if you take 630,000 and divide it by 8, that's the number of neurons that are in a single 1.5 by 1.5. And we're getting down to fairly small numbers of neurons, right? I mean, you know, there's a lot of neurons in the brain. We're measuring at a single point the combined activity of on the order of, you know, something on the order of 100,000 neurons, right? Which is actually a fairly staggering accomplishment. 3 by 3 by 3 millimeters, that's what it looks like. So there's a tip of an index finger with a little 3 millimeter square on it. So just so you have a sense of how small we're talking about. So it's pretty small. And these images are obtained over time, about 1 every 2 or 3 seconds. Right? And we'll come back in a moment about why that's the speed. So basically, the scanner runs, that's one picture of the brain, 2 seconds, and each acquisition is a 3-dimensional, right, volumetric picture of the brain, where the measurement at each point is one of these voxels, and the value inside that voxel is something that reflects the amount of oxygenated hemoglobin, right? Okay, so you get these pictures over time. Pop, 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 pop. So let me just give you a sense of what the data might look like acquired from one of these sorts of studies. So now I've shown just a single slice, and I've put a little box back in the occipital cortex here, where the visual cortex might be located. First stop for processing of visual information after the lateral geniculate nucleus. And let's imagine that we take a subject, our typical college student, and we pay him with like a Starbucks card, gift card, and we put him in the scanner. And what we're going to do is we're going to flash lights. So we're going to put him in the scanner. It's completely dark. And then at time 0, boom, we're going to turn on the lights and then back off again, right? So just a real big flash of light and then back off. And let's imagine not only do we make this functional neuroimaging measure, but we give him 2 Starbucks gift cards, and we actually also put an electrode in. So we get a direct measurement, like from, from the neurons at that site as well. It's a theoretical experiment. This has been done in cats, not in college students. But let's imagine we have these 2 measurements. Okay. So what you have is at time 0, boom, flash of light. It just on, off, real quick flash of light. If you had a measurement, every second of the neuronal activity from that point, right? So measured from the electrode, of course what you'd measure is from these V1 neurons before the light comes on. It's like tick, 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 tick. Light comes on, tick, 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 right? So they go back to being quiet. So you have this big barrage of action potentials and spikes that would happen in V1 neurons with a flash of light and then back off silent again. And you're also now, of course, measuring the bold functional MRI signal, which I will show in blue over time, that ensues following that flash of light. And it's something that looks like this. And what you notice, of course, is that first off, it is delayed in onset. So the change in the signal happens after a 2-second delay. It rises slowly to a peak after 5 seconds and then slowly back down over the next 5 seconds or so. Okay? And of course, this slow, gradual change in the imaging signal, right, in relation to the rapid change in neuronal activity is not surprising, because we got to deal with all this stuff, right? And in fact, and most of the delay happens at this first step, because changes in neuronal activity give rise to changes in the diameter of blood vessels. And you can imagine blood vessels, they're kind of like these springy, elastic things. They've got to dilate, and then it brings more blood in. So this takes time. It's a plumbing change, right? So sudden changes in neuronal activity give rise to these changes in plumbing and blood flow, which takes some time. And so as a consequence, changes in neuronal activity give rise to a bold fMRI signal, which in the parlance of engineers is a low-pass linear system. That is, low pass meaning that only slow changes in neuronal activity are manifest in the bold fMRI signal that you measure on the other side, right? So what's shown there at the top is, imagine an experiment where you kind of flick the lights on and off, right? So you have the subject in the scanner and it's like lights on, lights off, lights on, lights off, lights on, lights off, that fast, okay? And you do that for, say, 10 seconds, and then you leave them in the dark for a little while, and then you flick the lights again and then back off. Now, of course, you're measuring from the neurons. Every time the lights went on, tick, 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 and back off, they'd be quiet again. But what's measured from the bold fMRI system at the top is just the smooth envelope, right, of when the lights came on and off. The blood vessels can't keep up with lights on, lights off at that speed, okay? And in a similar way, another representation is just a single light on, light off activity gives rise to that shape down there. I'm not going to get into the great details about this, but it turns out that that blood flow response, right, that bold fMRI signal response that happens to a single spike of neuronal activity, in the parlance of engineers, is referred to as the impulse response function. So if any of you have an engineering or linear systems background, what that means is that, to make a long story short, in a low-pass linear system, in a linear system like this, if you know what the signal response is to a single little spike of neuronal activity, you can predict the output of the system to any arbitrary input, okay? This is what makes neuroimaging as an enterprise feasible, is that it's quite predictable and lawful how neuronal activity gets changed into a blood flow signal, okay? It's a very simple mathematical process which involves convolution, right, in linear systems analysis. Now again, of course, that does not mean we have any idea how instructions and stimuli and things get changed into neural activity. But once you get a good measure of neural activity, you can predict what the blood flow response will be quite clearly, okay? So that was a little expression of how in time, like a sudden change in neuronal activity in time gives rise to a change in blood flow that's delayed and dispersed over the course of about 5 seconds, right? How about in space? Well, if you could create a point of neuronal activity at one spot, right, that gives rise to a change in blood flow which is spread out, okay? So a point source of neuronal activity gives rise to a change in blood flow, right, a blush of blood flow around that point, which is about 3 to 5 millimeters wide. Okay? This is why when we make our measurements using functional MRI, we get pictures of the brain about every 2 or 3 seconds, and we get our little voxels, right, the little resolution that we get, at about 2 or 3 millimeters. And the reason is we're limited by the physiology of the system, right? It's possible we could run the scanner. It's easy. You could run the scanner and get a picture every 100 milliseconds if you wanted to. Right? And you could get those voxels down as small as you want. You can get them down to 100 microns, right? You can make them really small. That's fine. But it wouldn't benefit you anything because you're measuring a physiologic response that is itself smooth in time and space, right? You'd just be way oversampling a very smooth response. So that's why imaging is constrained not so much by the technical limitations of the scanner, but really by the physiologic properties of the signal we're measuring. We can't measure anything smaller, we can't measure anything faster, because that's all the blood flow will allow us. Okay? Okay. So now so we've talked then about some of the properties of the system too, and sort of its resolution in time and space. Now let's finally talk about cognition, right? So let's go through a prototypical experiment. Let me just describe how it works in neuroimaging and sort of the inferences one might make from it. And this will start to now leak into some neuroethics kind of content. Okay, so here is like a typical kind of brain picture. What I want to do is work our way up to how we end up with this, okay, from a neuroimaging experiment. And so this seems pretty innocuous. It's a 3D cutaway view of the human brain. You can see the face, kind of snazzy. There's like little blue dots on there, which we're going to learn exactly what those are, and there's something about neural activity. Well, here's actually what that picture's from. That picture's from testimony that was given at the mitigation phase, or I'm sorry, at the sentencing phase in defense of and in support of mitigation for this guy, a guy named Brian Dugan, who was like a really brutal, horrible serial killer guy who ended up killing a cop in Illinois, was arrested, sentenced, convicted, and at his trial, testimony was provided in his defense by a guy named Kent Keel. And Kent Keel has a research operation which involves driving a scanner, an MR scanner, in the back of a tractor-trailer truck and driving it into the exercise courtyard of prisons and penitentiaries in the American Southwest, and then doing imaging studies on criminals. And he's doing really interesting work trying to figure out like, oh, well, like what's the difference between criminal brains and not criminal brains, and are there any differences? And he's quite interested in particular of this behavioral phenomenon of psychopathy, right, which is a tendency for people to have very little emotional response to the suffering of others as a broad statement. And so Kent wanted to testify that after putting this guy, Brian Dugan, the convicted killer, in his scanner and making some measurements, which we'll go into in more detail, he found that there was a neural signature, there was a brain activity signature, which indicated that Brian Dugan was a psychopath. Sorry, Kent. So Brian Dugan was a psychopath. That was the claim, that this brain imaging picture let Kent know that Brian Dugan, the defendant, was a psychopath. And I'm not sure why that was actually supposed to be a mitigating circumstance for the death penalty or anything else, but that was how it was raised. Okay, I'll leave that to a lawyer to figure out. So what's behind that image? Well, the specific claim was that psychopaths show reduced responses to emotional stimuli within the amygdala. Okay, so one of those blue dots is in the amygdala, right? The amygdala is, this structure that deep within sort of a subcortical structure just anterior to the hippocampus, often implicated in the processing of emotional stimuli. I'm sure there are probably some of you in the room who know much more about the amygdala than I do. But in any event, so the claim was that psychopaths show reduced responses to emotional stimuli. And in fact, the particular experiment was something like this. You take a subject, you put them in the scanner, and over time, you show them blocks of stimuli of faces that are either contain emotion, in this case a fearful face, or an ostensibly neutral face. Actually, this face looks kind of angry to me. But anyway, an ostensibly neutral face. So you show faces over time, fearful face, neutral face, fearful face, neutral face, and you measure the blood flow signal, right, the bold fMRI signal, from a given point in the brain. So that little black dot up there is in the amygdala. It's not really, but close enough. And then that's the bold fMRI signal rising and falling from that point in the brain while these stimuli are here. And if that's the data you had, like, this looks pretty good, right? Like every time that kind of scared looking face comes on the screen, the signal goes up. When the neutral face comes on, it goes back down. That looks pretty good, okay? So what you can do is, since we know the shape of the blood flow response and all the rest, we can actually fit it with a model and say, okay, if it was the case that there were neurons that increased their firing whenever the fearful face came on, this is exactly what the blood flow response should look like. It matches pretty well, right? We could then quantify that impression, like, okay, well, how good was that fit? And there's lots of different statistical ways to do it, but effectively what we're doing is we're taking the average of the time that the fearful faces were on and the average of the time the neutral faces were on and asking how big or reliable the difference was between those two things, right? And that then is a value, like a statistical value. Maybe it's a p-value, maybe it's a t-statistic, whatever it is that's been measured, or a difference or percentage change in signal. And that is the value that you calculated from that one point, which is indicated in red in the brain. Okay. Now, what you could do, of course, is just like write down on the image, like you'd write down, well, p equals 0.002, right? You could imagine writing that down in that little box, but of course, that would be cumbersome. There's lots of points you want to look at. So let's color code it, and we'll call a small effect kind of red, medium effect orange, big effect yellow. So you just create a little color scale, right? That's standard. But then, of course, you do that calculation not just for one point in the brain, but for every point in the brain, right? So every point gets a calculation, and then you stick a color in there, indicating the statistical effect that was present at that location, right? Okay, so that's the basic level of analysis. This refers to as a univariate analysis. In a moment, when we come back at the very close of these lectures, I'll say a little bit more about like what's happening nowadays involving multivoxel pattern analyses and other sorts of more multivariate techniques. But this is a univariate, what's referred to as a massively univariate analysis of the statistical effect of your stimulus manipulation at each point in the brain. Okay. Here's where we get into the domain of neuroaesthetics, which is how we go from this kind of crummy-looking, blocky digital picture of the brain to something that's really smooth and pretty and is worth putting on the cover of Time magazine. Actually, is Time still in business? Putting on the Time website or presenting in court. Okay. So this is, of course, what we start with, right? So 3mm voxels, all these little points, all the rest. Well, one obvious problem that we need to deal with right off the bat is that even though we've assigned a statistical value to every point in the brain, clearly there are many statistical values there that are not significant, right? That if you run some calculation and then force each point to have a value, every point will have a value, but a lot of it could just be noise, right? It has nothing to do with the faces. So that means then that what a statistician would tell you to do, a frequentist statistician would say, well, adopt a threshold. Say, what is the amount of evidence that you require to claim that a point in the brain actually did follow along with the face stimuli? So you adopt some threshold. Ah, that's better, okay? And in fact, that threshold, there are very well-established statistical techniques for picking a threshold that controls the false positive rate of these dots in the brain, okay? Very, very well established. There's really, if you want to do this properly in a neuroimaging study, there's absolutely no reason to do it wrong. So, no, there's bad studies out there that still do it wrong, but there's absolutely no reason to do it wrong. Anyway, so that can be done. You apply a threshold. Okay, good. Now we're seeing those points which are just kind of above that threshold. Already the map looks quite different. But here's the next thing. It turns out that there are very good signal processing reasons why smoothing the data in space could enhance your ability to detect stuff. Right? So if you take your data and you apply like a little bit of a digital blur to it, it enhances your ability to detect sort of coherent areas of response. Now, at the expense of detecting small areas of response, but if you blur your data a little bit, you can see the larger areas of response will become more significant and easier to detect. And again, this is like a very well-established thing in signal processing. If you want to detect a signal of a certain size, you should pass your data through a filter of a certain size and all the rest. But so it's quite common to provide some digital smoothing to the data, right? Now, this has the consequence of enhancing the appearance of blobs of activity of a certain size and erasing or diminishing other size blobs, right? So, but it's a trade-off, you can only detect so many things in an experiment. You want to detect some of the bigger stuff, so you do some digital smoothing. Okay, that looks pretty good. The problem, of course, now though is like, I can see where that blob of colored activity is, but it's hard to figure out where in the anatomy that's located because the underlying picture, right, the source images were really kind of blurry. Here we go. Actually, at the same time I ran this experiment, I also collected one of those crisp, sharp T1 images of brain anatomy. I've got that lying around. How about I display that blob of activity on top of that? Okay, good. Now that's much better. Now I can really see where that blob of activity is in relation to the structure of the brain. But I've got a final problem. The brain's a three-dimensional structure. There's lots of slices. And how am I going to display this? Oh, no, I'll get my friends over in computer graphics to do a 3D cutaway view, which shows the activity kind of as a 3D rendered. Okay, great. So now every step along the way, perfectly justified. But the distance that we've traveled from the raw data to the final version of the picture that we present is enormous. And I would argue, actually, that there's a certain aesthetic that people who do neuroimaging work adhere to. There's a way that these brain pictures are supposed to look. And even though there's lots of valid reasons for making these statistical judgments, nonetheless, there's a lot of different judgments one could have made. And the ones that tend to be made are ones that serve a certain kind of aesthetic. And certainly that's been true for the last 20 years. I think it might be changing a little bit now, but that's certainly the case, that brain images have to look a certain way to be published. Okay. So, all right, well, that's got us through the data for one subject. But now suppose we want to make this measurement not from one subject, from a bunch of subjects. Well, then we have to deal with this problem, which is that different subjects have different shaped brains, and different subjects might have different patches of activity, right? So here's the results from 10 different people. And different people, different shaped brains, blobs of activity. It's like, all right, the general story looks pretty good. Like most people have a blob somewhere up front there. That guy, it's weirdly split in two. That one's a little farther back. But I want to kind of get what the average sort of statement is across a group of people. So to do that, I'm going to average the brains together. I can use digital warping techniques to put all the brains into a common space and average the data. And then I end up with something like this. Okay, so that's sort of the typical thing. So now we come back to then this original claim, right, which is that we wanted to claim that psychopaths have a reduced amygdala response. And so that data might look something like this, right? This is not actually Kent's data. This is simulated cartoon data, but it's something like this, that if you took 10 psychopaths and put them in the scanner and 10 normal people, and you looked in the amygdala, right, from all those people, and you measured what was the average blood flow response or average statistical effect or average effect size, whatever it is you're going to measure, in 10 control subjects walking on the street and 10 psychopaths, and those are the error bars, which I've just expressed as a range, you might observe something like that. Okay, and if you got that data and who knows what the standard error bars are like, that might actually work out, right? That might be a valid claim. The psychopaths actually do have a reduced effect. Of course, and as everybody in this room knows, it's a very different claim from this claim, which is this guy is a psychopath, right? And the reason it's, of course, a very different claim is because that guy, the Brian Dugan guy, you might have measured this, where the arrow is, right, on the distribution of responses from the psychopaths and the normals, and you'd say, well, yeah, no, it's consistent with being a psychopath, but there's enough overlap, right? And so I say this, everybody in this room is quite sophisticated and knows this kind of stuff, but this is clearly something that neuroimaging data doesn't get to sidestep, right? Generalizing from populations to individuals is just as hard in this work as in any other. Okay. So that prototypical design that I've just described for you, I think is a fair description of like neuroimaging 1.0, right? So that's kind of how it's gone for like the last, the majority of the last 20 years. That's what neuroimaging experiments have been like. These sorts of designs that have the property that there are blocks of time where one stimulus or another is presented, and you measure the difference between those points, and that's the sort of stuff you do. In a few minutes, I'll talk a little bit more about neuroimaging 2.0 towards the end of the talk, but this is sort of how most of it's been done. And just some key things to take away is that these are signals which are, of course, an indirect measure of neural activity. It's, you're measuring a bulk activity change, which I'll unpack in just a moment. And this general design is called a subtraction design. So the idea is one wishes to measure what is the response to the emotional content of faces, and to do so, you compare emotional faces to neutral faces, look at the difference in brain activity between those two, and then attribute the activity that you see, the difference in neuronal activity, to the difference in the stimuli, which in this case is the presence of a fearful or emotional expression, right? So the core idea of a subtraction design. And I just want to touch on a final couple things that can sort of interfere with the conclusions that one may make about these sorts of studies, particularly when you start looking at populations. One thing to keep in mind is that the bold fMRI signal, this thing, has no direct absolute interpretation. It's actually a unitless signal, right? Because it's something that has to do with a time parameter, a fit to a time parameter of that T2 decay curve. It's a unitless number. And so it can be quite hard to make a comparison of that just absolute signal magnitude between one person and another, or one population or another. And there's lots of things that can affect that signal magnitude besides the change in neuronal activity that I've mentioned, right? So lots of stuff like, people who have more dental work, right, which is metal and is going to alter the local magnetic field and then is going to alter the signal from that area, or people who might have more or less iron in their blood, or people who are smokers, or have higher or lower concentrations of hemoglobin. All of those things will alter that absolute level of the signal. So it really, the fMRI signal can only be interpreted in terms of a difference between two states, right? You can't actually just take the raw amplitude of the signal and make a meaningful statement about it. Of course, and this is something that, again, this group would be quite capable of understanding, is that we're measuring a hemodynamic proxy of neural activity, where we're not directly measuring neural activity, we're measuring this blood flow response. So anything that messes with that system 2 coupling is going to mess with our measure, right? So if for some reason you wanted to compare two groups of people, and one group had a systematically different pattern or form of blood flow response to a given change in neuronal activity, you could mistake a difference in blood flow coupling for a difference in neuronal activity between the groups. There's lots of ways you can get this wrong, right? So if you just study old and young people and try and make a comparison of the level of, so you take old people and young people in the scanner and you show them these pictures of emotional faces and neutral faces, you might discover that, wow, my God, old folks don't seem to react at all to emotional pictures of faces. They must be psychopaths. Well, actually, it's just that they have reduced neurovascular coupling, right? The degree of blood flow change that you measure for a given change in unit change of neural activity is reduced. There's just a change in physiology with age. Lots of other stuff can affect this: drugs, hormonal states, gender, lots of stuff. So as a consequence, one has to be very careful when you make these sorts of group comparisons. All right, and then just finally, something that is well known in the field but not talked about enough is that we do our studies almost always on young, healthy college students. And if one wants to make claims about an incarcerated population, or people who grew up in abject poverty, this is quite different from the typical Penn undergraduates that are the bulk of people that we study. And there's lots of reasons why the particular results we obtain from them might be different. Okay. The final thing I want to touch upon, and this will be the final point before we take another break and then go into inference, is just something about the sort of getting down to the fine scale limits of what we can measure about neural activity with this stuff, right? So the experiment that I talked about, right, has the property that there's a bulk level of greater neural response to the emotional faces versus the neutral faces within the amygdala, right? All the neurons in the amygdala, or at least a fair number of them, fire more avidly for the emotional face than they do for the neutral face, right? That's what made that study possible. So another way of showing it in sort of this cartoon form is that those little gray circles represent a population of neurons in the amygdala. And what we did was we showed the neutral face and the fearful face, and all the neurons just brrr, brrr, they fired more, and then the blood flow increased, and boom, we had a measurement. Here's what wouldn't have worked, right? Which is, let's imagine you have a region of the brain or a patch of cortex that has the property that, the pattern of neuronal firing differs between the two, but the average bulk level of firing is the same, right? Undetectable by this experiment, right? And you can think of tons of sorts of representations, right, which have the property that they're encoded by the pattern of neuronal firing at a fairly small scale cortical level, which would be undetectable by these techniques that I've described so far. There are other ways of doing these experiments. That's where we get into neuroimaging 2.0, where you take advantage of sort of, for example, the fatigability of neurons. And you start like repeatedly presenting stimuli and now look at the degree to which neurons fatigue over time or habituate to repeated presentations of stimuli, which turns a pattern difference into an amplitude difference and then is measurable, as well as multivoxel techniques, which I'll talk about at the end. But just to be aware, this, in order to measure something with bold fMRI, you need to produce a bulk change in neuronal activity in that 630,000 neurons, which make up the voxel that you're measuring from. Okay? And unless you can do that, you're not going to see the difference, which is why there's all sorts of stuff that you could present people with. I put someone in the scanner, I show them pictures of bagels, I show them pictures of donuts. You know what a bagel is, you know what a donut is. They're clearly different mental states. But I look at the brain activity map from a bold fMRI study, no difference, right? So obviously there's lots of ways to get false negative results if the information is encoded in a pattern and not bulk.